All right. The final talk of the session, because the final talk of the session was supposed to happen by Heather Machado, was canceled. So the final talk of the session is by Chuan Li, and then after that, Mike Lynch will want to say a few words.
Okay. Good afternoon, everyone. I'm Chuan Li from George Zhang's lab of molecular and genomic evolution from the University of Michigan. Today, I'm going to introduce about the fitness landscape of a tRNA gene. So fitness landscapes are important because it can inform us on the strength and direction of selection, and also it can reveal possible evolutionary trajectories. A previous study has found that among the 120 trajectories leading to 5 mutations, only 18 of them are accessible. Although they are very important and informative, describing fitness landscapes is a formidable task because of the huge genotype space and complex epistatic interactions. Take a 6-9 base pair variable region as an example, which is the length we use in this study. It will be one wild type, 6-9 times 3, which is 207 single mutants, over 20,000 double mutants, 1.4 million triple mutant, etc. Moreover, in previous studies, many other metrics other than fitness were used. For instance, people have used in vitro selection to study binding affinity landscape, record the chemical readouts to study the activity, the catalytic activity landscape, and also study the landscape of other protein functions, such as GFP fluorescence. So for this project, I decided to directly measure the fitness. So I focus on the fitness landscape of a tRNA gene in yeast, which is a 72 base pair gene, a single copy that code for arginine with anticodon CCU. Here, I use precise gene replacement and barcode sequencing, where the tRNA gene of interest also serves as a barcode to simultaneously track the fitness of many tRNA variants. Specifically, I measure fitness by tracking the barcode frequency over time. So here is the experimental layout. So at each site, I have 95% as the original wild-type nucleotide. And for the other 3 nucleotides, I make 1% of each. This way, I can maximize the fraction of double mutants. So as a result, 27% will be double mutant. And then I link these tRNA variants with a marker gene, Euro3 gene, and use that to replace the LU2 gene originally inserted at the genomic location. And then I collect 100,000 colonies at room temperature. So room temperature is great in this circumstance because at room temperature, the wild type and deletion grows very similarly, so which allow a very fair representation of these variants. And then the whole pool of colonies was subjected to competition under high temperature challenge. And then the pool before and after competition was sequenced using pair and Illumina sequencing. Because this tRNA is very short, only 72 base pairs, so we can cover it by a single read. So we can do it from both ends to ensure high quality of sequencing. So using that information, I can further estimate the fitness of this variant and do further analysis on epistasis and the underlying factors determining fitness. So here is the result. This figure shows the average fitness upon mutation. So there are 69 variable sites. We expect to see that there are some sites is important For instance, the anti-codon is important because it determines the recognition of codon, and also this tRNA is driven by internal promoter. So these sites, of course, are important. Indeed, we see that among the 9 sites that have average fitness dropped below 0.75, we have all the 3 anti-codon positions and 5 nucleotides from the internal promoter. Also, fitness values can help us predict evolution. To prove that, I look into the evolutionary history. So I aligned 200 non-redundant eukaryotic tRNA genes to this tRNA gene, actually, and requiring that the distance between any 2 tRNA sequences are over 10%. So for the left panel, let's look at the 69 sites. So if the mean fitness upon mutating a site is low, meaning the fitness defect is large, we would expect that the site need to be conserved. Indeed, we see that there's a high fraction of sequences identical to yeast at that site. Moreover, on the right panel, we can look over the 207 single mutants. So for the mutants that appears in phylogenetic data at varying frequencies, they are in general acceptable. So we expect that the fitness of the single mutants are close to 1 because they are acceptable. So for mutations that rarely appears or never appear in evolutionary history, we would expect them to have fitness defects. So indeed, you can see that there are a lot of dots in the button, which shows varying degree of fitness defects. So here is the overall distribution for single, double, and triple mutations. So on the left side, you can see that for single mutants, many of them had fitness close to 1, and 42% are significantly deleterious, and 1% are significantly beneficial. And for double mutants, the middle panel, we can see that the fitness is drastically shifted to the left, meaning they have some fitness defects. And the right panel is for having 3 mutations. We can see that, so they are almost not growing because 0.5 means no growth. So I get all the single mutants, 61% of all the possible double mutants, which is 12,000, and 1.6% of all possible triple mutations. So in total, I get 65,000 variants for this tRNA. Further, I want to do some validation of my fitness measurement. So I use 3 different metrics. So the left panel, I did pairwise competition with a reference strain, followed by flow cytometry. And the right 2 panels, I used a single strain growth curve and looked at the maximum growth rate and proliferation efficiency. And we can see that the in-mass competition measurement of fitness agrees pretty well with the other metrics. So having fitness for both single and double mutants allow us to quantify epistasis. So I was able to quantify epistasis between almost 13,000 pairs of mutations. Here I define epistasis as FAB minus FA times FB, where FAB is the fitness for the double mutants, and FA and FB are fitness for the corresponding single mutants. So as you can see from the distribution, I think I have a background color that wasn't shown very well. So for all the 13,000 pairs, so a larger fraction is showing negative epistasis. So here the purple bars show the significant ones that agrees well between the 6 biological replicates. So among them, 86% showed negative epistasis. However, we would expect that the pair site shows a different pattern because different Watson-Crick pairs may be functionally similar. So here I show two examples. So if site 11 or site 23 was mutated alone, there will be a huge fitness decrease. However, when you mutate both sites at the same time, the fitness is restored. Similarly, you can see the pattern also holds for site 51 and site 61. So when we look at the overall distribution, you can see that when we switch from one Watson-Crick pair to another Watson-Crick pair, fitness are largely positive. It is not all positive because this tRNA is pretty short. So actually, apart from structural purpose, every sequence also have other purposes. And this is switch from Watson-Crick pair to non-Watson-Crick pair. And actually, both groups were significantly more positive than other sites. Moreover, we want to further look at the underlying mechanisms determining fitness. So what are the factors that can determine fitness of a tRNA gene? For example, it should have correct folding, adequate amount of expression, and also other sequencing features to attach the amino acid, bind the ribosome, and recognize codons. And also the number of mutations that may also matter a lot. So here I focus on tRNA folding as a determinant. So I try to predict the proportion of functional tRNA molecules, which is drawn on the y-axis, x-axis here, with two criteria. First, they should have the correct folding to the functional secondary structure. And second, they should have the correct codon, so that they are recognizing the correct codon. So using that criteria, we can see that when the proportion of functional, predicted proportion of functional tRNA molecule is higher, the tRNA has higher fitness, although with a diminishing return. And after controlling for the number of mutations, the trend also holds. So for single, double, and triple mutants, we can see that fitness is highly correlated with the predicted proportion of functional molecules. And the correlation for the raw data, for the 65,000 data, is 0.4. So in conclusion, combining precise gene replacement and barcode sequencing, we get to estimate fitness for over 65,000 variants. And epistasis has a strong negative bias, except when the mutations occur at Watson-Crick paired sites. And thirdly, fitness is correlated with the predicted fraction of functional tRNA molecules, thereby revealing a biophysical basis. Finally, I would like to thank people in my lab and Professor Kutler for discussion and suggestions. And also, this project is supported by NSF DD grant and my advisor's NIH grant. So, and also, thank you very much for your attention. And I'm happy to, I'm happy to answer any questions. And also, if you want to check more, this paper is published 2 months ago on Science.
Plenty of time for questions.
Missed it, but did you see any difference in the average fitness for mutations at the GC pairs and the AT pairs in the regions that form pairs?
Oh, so that figure I showed had both the GC-AT, or like it can either be the same type of Watson-Crick pair or different. I think for the ones I observed, actually the same type of switching might be better, but because I think the molecule is so short that many of the GC-GC switch doesn't work. Okay. Did I correctly answer that question?
I was more wondering if you have just one mutation and then you break the pair, and if it was a GC pair to start with, or if it was an AT pair, do you see a difference in the average fitness effect?
Sorry,
I don't know if you see what.
Yeah, we can discuss that
okay.
afterwards. Sorry.
I have a question.
Yeah.
Have you observed a substantial fraction of advantageous mutations?
I think for...
Maybe you could just repeat the question.
Yeah, so the question is, did I observe some examples of beneficial mutations? So I think among the 207 single mutants, I find 2 that are significantly beneficial. And because these ones have the highest read number, so it's very I think very likely to be true, and it's also true across 6 biological replicates. And there are also many cases, some fraction of cases observed for double and triple mutants. Yeah, so indeed, some of them tend to be beneficial. However, this is under high temperature challenge, so we don't know how well they will do under 30 degrees Celsius or natural environment. Thanks.
So unfortunately, Heather Machado's talk was canceled, but Mike Lynch wants to say a few words.
Yeah, I just wanted to say a brief word about the Jim Crow Award. Several people have come up and asked. So this is not an award funded by the GSA directly. We're funding it. There have been a couple anonymous donors that got things off the ground, but we don't have a whole lot of money in the pot to keep this going. And as you heard Dan Hartle's talk the other day, Jim Crow is one of the most wonderful, he's certainly the most wonderful human I've ever met, probably the most wonderful vertebrate I've ever met, or eukaryote, really. He was just an incredible guy, and he's had an influence on an enormous number of people. You can, if you wish to make a donation, you should be able to find, I've got the fine print up here, but you can send a check directly to the Genetic Society of America. You can find the address on the GSA webpage. If you have further questions about it, you can ask me. If you're willing to donate $25,000 or more, we'll throw in a viola, I think, and with a D-A-G-C set of strings. So my son's a cellist. He knows what the dumpsters are, where they're all stored. So anyhow, you can talk to any of us, but I know several people are interested in making such a donation, and we'd certainly like to keep the award going because we'd like to be able to support the travel for the finalists for this award in the future, assuming the meeting is held in the future. So that's all. Okay.
So let's thank all the speakers, and